Welcome back to the Classics of Immunology Journal Club. And this is going to be an inaugural journal club, a real-time journal club, discussing papers that have just been published to see how they fit in and to whatever you happen to be interested in, whether then we can also in real time find out whether they stand the test of time. Before we get started, don't forget to go on the YouTube channel and like the video and subscribe and click on the bell to get notified for more. Check out my website. You'll find all kinds of information there, and including many papers from about all the classic things that have happened in immunology in the last 200-300 years. So today we are going to review a paper that came out in Science and on December the 16th of 2022. And the first author of this paper is Greg Allen, and then there's 14 more authors, and then the last or senior author is Wendell Lim. And the title of this paper is Synthetic Cytokine Circuitry that Drive T cells into Immune-Excluded Tumors. The background on this paper is that this is a biological engineering paper. You know, we had genetic engineering or molecular engineering. Well, now we've got a whole cadre of scientists that are trying to take cells and re-engineer their genetic expression so that they can get them to do what they want them to do. And so one of the burning questions in immunology today is how can we make T cells kill tumor cells better than they have been so far? So the background on this particular paper is that it's about interleukin-2. You might expect that I might go into that subject, because almost 40 years ago now, Alice Reinhartz and I published a paper and first reported that the way the T cell immune response works is that the antigen-specific T cell recognition through the T cell receptor leads to an antigen nonspecific T cell response that's mediated by interleukin-2 and other interleukins and cytokines. So the T cell receptor is a recognition element, but the work of the T cell is being done by cytokines. And so these people have figured out that that might be a way that they could re-engineer killer T cells to do their job even better than they have been so far. Now, it's also important. One of the things in this paper that I didn't like or appreciate was that they called interleukin-2 an inflammatory cytokine. Now, I disagree with that. Interleukin-2 is a regulatory cytokine, and that's an important distinction and difference. What interleukin-2 does, and we and others subsequently showed that's what happens with interleukin-2 in terms of its biology, is that it initiates proliferation of the antigen specifically activated T cell, and at the same time it also, or simultaneously, initiates differentiation of the T cells so that it takes on effector functions. Probably the most well-known effector function of a T cell is to become a killer T cell. Also can become a helper T cell. And those two are not necessarily exclusive of one another. That's another subject right now. So the introduction of this paper is they start out by saying that in terms of the engineered T cells, the so-called CAR T cells, chimeric antigen receptor T cells, so that they have the antigen recognition component of an immunoglobulin on the outside of the T cell and the activating signaling portion of the T cell receptor on the inside to activate the T cell. So they call it a chimeric antigen receptor T cell. It seems to work fine or pretty well for liquid tumors, but for solid tumors, they've had problems, and they often fail to respond to these CAR T cells, and they think that it's due to several different factors. One is the microenvironment of a solid tumor is different than the liquid tumors, and it seems to block the ability of these chimeric antigen receptor T cells to really function well. The first is that it seems to inhibit or prevent the infiltration of the solid tumor by the CAR T cells themselves. Second is that there are molecular mechanisms in place that turn off the activation mechanism of the T cell receptor. And third, as a consequence of that, it turns off the effector functions of the cytokines, interleukin 2 particularly, because then you don't get clonal expansion, proliferation, and you also do not get differentiation to a killer T cell. And the differentiation of cytotoxic T cells has now been well worked out over the years to be due to the granular mechanism. Inside killer T cells are granules that are chock full of all kinds of really neat molecules, one being the molecules that encode perforin, and which pokes holes in the membrane of a target cell. And then there's also all kinds of proteolytic enzymes that are called granzymes that are then secreted through those holes into the target cell and digest that target cell from the inside out. It's a pretty fail-safe mechanism of killing other cells. That's all stimulated by interleukin 2, by the way. The genes and so forth that have been identified and isolated are, for the most part, most of them or a lot of them are due to STAT5 induced genes. IL-2 activates STAT5, and that's the way the whole system works. So the question is, what to do to try to counteract these suppressive mechanisms. And of course, what I've published on and what I've said over the years is that all these mechanisms for fine-tuning the T cell effector response are essentially they're negative feedback loops that are activated at the same time that the activation situations occur. So the rationale that these people took in this paper was they said, well, this interleukin 2 therapy in cancer, a la Steve Rosenberg from the NCI, is quite toxic. And the reason that that's the case, this is a Kendall parentheses, is that Rosenberg gave IL-2 as an IV push with a syringe into the vein every 8 hours, and he gave gargantuan doses of interleukin 2, so that the whole total body level of interleukin 2 was sky high, and consequently, one of the things that happens when that occurs, when the concentration goes up, is that you not only activate the so-called high affinity trimeric interleukin 2 receptor that occurs on activated T cells, antigen activated T cells, you also then activate, which is the alpha beta gamma receptor. You also activate then cells that express interleukin 15 receptors, because interleukin 15 receptors are constructed with an interleukin 15 alpha chain, but the beta chains from IL-2, the beta and the gamma chains from IL-2. So that the beta and gamma chain will still bind interleukin 2, but they bind it with about 100-fold lower affinity, meaning you need about 100-fold higher concentrations. But of course, Steve Rosenberg accomplished that by squirting in as much as he possibly could. So because of the toxicity, though, poor interleukin 2 has gotten a bad name in oncology, and that gave the rationale for these people to see if they could circumvent that in some way. The toxicity that occurs when you do that is that you get the so-called cytokine release syndrome, because what happens is that is secondary to capillary leak, which is secondary to the real pro-inflammatory cytokines, interleukin 1, interleukin 6, and TNF, and they cause the capillaries to leak all over the body, essentially. But these people thought maybe if we dysregulate the interleukin 2 production, or the genetic turn on of interleukin 2 in the cells, so that the T cell receptor pathway is not the one that's activating the interleukin 2 genes, maybe we could get around all this toxicity, and we could decrease the negative feedback that occurs. The negative feedback is really occurring through the expression of CTLA4, PD1, and a lot of these inhibitory molecules that occur, which are all also interleukin 2 induced. So if we dissociate the T cell receptor pathway in activating these negative feedback loops, that might help things. And so what they did was they put the interleukin 2 aspect onto this system through a different receptor, not the T cell receptor. They used a synthetic Notch receptor to drive interleukin 2 production. which does not activate the negative feedback loops. Let's go to screen sharing now, because what I'm going to show you is a figure from the summary of this paper that essentially summarizes the significant findings. And this paper, like so many of the papers that are come out daily and weekly in modern science, this paper is a very complicated paper. And if you're interested in genetic or biological engineering, I think you should read the whole paper very, very carefully. So the title of this figure here is a therapeutic T cells engineered with tumor-triggered IL-2 production, infiltrate and clear immune-selected tumors. So they have a CAR T cell that it will recognize a molecule on the surface of the tumor, and they have engineered in the same CAR T cells a secondary way to activate interleukin-2 production. So here's one of the control groups of the mice being treated with systemic interleukin-2, and that's by comparison to cell-derived cytokine interleukin-2. So you have a CAR antigen receptor, chimeric antigen receptor, and then you have synthetic notch driving interleukin-2. So the T cell through the CAR will recognize the tumor. And this is also a tumor-specific recognition system that will then tie in interleukin-2. And in the B portion of this figure, you can see that the CAR T cells infiltrate these so-called immune-excluded tumors. This is the CAR only, and this is the CAR plus the IL-2 circuit here that we have described in this cartoon right here. And you can see that in this histochemical micrograph, you can see the infiltration of the tumor with the CAR T cells. And then in C here, we see the CAR T cell tumor clearance. We've got tumor volume on the ordinate and the days after the injections of animals. So here is the minus IL-2 control, and this is plus the IL-2 circuit. And you can see that the tumor volume drastically decreases, leading to essentially survival of these animals by comparison to the control. So that's all I want to show in this paper now. And the discussion in this paper is, or the critique in terms of this. So the authors of this paper end up with a hypothesis that these IL-2 circuits are effective because they bypass the CAR T cell antigen receptor to provide IL-2 in a more potent autocrine configuration. But they don't really define what that is. And I'm very skeptical of that whole thing. My critique of this paper is that, well, we should watch this. This is a whole new area of scientists for these biological engineers to see whether or not they can get the body to do things that they want it to do, rather than what the body's has been evolved for and selected over the millennia. So what they're trying to do is they're trying to improve upon Darwin. That's what this paper is all about. The thing is, the authors assume that they are bypassing the T cell receptor dependent negative feedback of the TCR signaling of the IL-2 gene. But the problem is that they haven't been able to easily measure and monitor the IL-2 receptor density on the cell surface in vivo, or the in situ IL-2 production. So it's a black box. They don't know what's going on in there, even though they have these very pretty cartoons to tell you what they think is going on. And this really gets to the point of what I said in the very beginning, and that is, the question is whether or not what we're dealing with here is a hormonal mechanism whereby we have a circulating molecule that is secreted into the milieu that then binds to a receptor, and that the whole response to the hormone IL-2 in this instance is dependent upon only three variables. One is the IL-2 concentration, the other is the IL-2 receptor density, and the other is the time that these two molecules have to interact with one another. Now, this paper was published in the same issue as another paper, another biological engineering paper that attempts to manipulate the timing of the activation through the T cell receptor and the activation via interleukin-2. And so I think you ought to read both of these papers. In addition, there's an editorial by an old friend of mine, Gregoire Altin-Bonnet from the NCI, who explains everything in a page or two. So that's another good way to delve into this paper, which is what I think you need to do in order to get on board with this biological engineering. So I'll stop there. Don't forget to go on the YouTube channel and like the video and subscribe, click on the bell, and check out my website. Thanks so much. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.